Welcome. Welcome. This is the last I3 session of the year. It's not like the it's a little bit special. It's going to be about non-traditional species, and it's going to be also about really the advanced assisted reproductive technologies that are developed for biomedical science, conservation research, and this is very exciting. We have three very prominent speakers to talk about that. So my name is Pierre Comizzoli. I am a research veterinarian at the National Zoo and Conservation Biology Institute at the Smithsonian in Washington, D.C. I've been working on wildlife reproduction for a long time. And I'm joined today to moderate this session by Matt Pettit, who is the Chief Scientific Officer at Mature Technology Limited. It's a barcode system for fertility centers. But importantly enough, for this session, Matt serves as a trustee and scientific advisor for Nature Safe, a conservation initiative preserving live cells for endangered species in collaborations with many zoos. So what's really important for all the participants to remember is to please put your questions in the Q&A section, not in the chat. Please, everybody, please, if you have questions to the speakers, please put them in the Q&A. They will be picked up by our moderator behind the scene, and we will be able to, as possible, to present them to the different speakers. So without further ado, I think we're going to be starting with our first speaker. Our first speaker is Andrew. Andrew, are you with us? So as you are getting ready, Andrew, to share your slides, let me introduce you. Andrew Pask, if you don't know him yet, is really the head of the Devo Repro group at the University of Melbourne in Australia. He's a scientist using comparative mammalian genetics to identify critical and conserved networks driving key processes in development. His work has led to the sequencing of several genomes, especially in marsupial species, including the extinct Tasmanian tiger. And actually, Andrew has established the Thylacine Integrated Genetic Restoration Research Lab, the Tiger Lab, which is a pretty cool acronym, to examine really the possibility of having these species back, the extinction of that species, as well as, especially important, to develop really the next generation tools to preserve and conserve current endangered marsupial species. So Andrew, welcome, and please, the floor is yours. You have about 20 minutes to talk. So what's going to be special with Andrew compared to the other speakers is that we're going to keep him a little bit longer after his talk for Q&A, because Andrew unfortunately cannot stay with us for the whole webinar. But please, Andrew, welcome.
Thank you very much. And thank you for the invitation to present at this incredible conference today. It's great to see so many people online from so many different countries, and I'm really excited to tell you about the work that I'm doing here at both the University of Melbourne and with Colossal Biosciences, which is a big biotech firm, which is really in exploring the possibility of de-extinction and the way that it can really change the conservation landscape. So that's what I'm going to talk to you about today. The reason I've been so passionate about this for so long in my career is because in Australia, unfortunately, we have the highest rate of mammal extinctions of any country in the world, which is crazy when you think of what a wealthy country Australia is. There's really no excuse for being at the forefront of mammalian extinctions. But at the moment, we have over 20% of our land mammals threatened with extinction. And at that rate, we're predicted to still have 1 to 2 extinctions every decade moving forward. And this statistic doesn't take into account adverse weather events, which are particularly problematic in Australia. We're having far more of these events, and they're of far greater intensity. And these are things like the horrific bushfires that we've had. We had some about 3 years ago that were really devastating, and big floods as well. We go from one extreme to the other, as we're experiencing in so many countries across the world now as a result of global warming. And what this does is any single one of those events can completely, eradicate a species and make a species extinct. Or even when it doesn't do that, it can reduce the number of animals in that particular species to below a critical mass, where they start to enter what we call an extinction vortex, where there just simply isn't enough genetic diversity left in that remaining dwindling population to really have a healthy and sustainable population bounce back from that again. So these are things that are really profoundly impacting, particularly Australia, but obviously these are massive global issues, and adverse weather events are likely going to really push that statistic of how many extinctions we're going to see, a lot faster as well. And I'll talk about how the technology we're developing for de-extinction can really help mitigate that as we move forward, because I think it's really, really important. So what my lab has been doing then is really trying to think about these next generation conservation tools that we need to develop to try and stop this from happening, basically. So we know that our current conservation toolkit is not enough. We're still seeing all these animal extinctions, so we need to be able to figure out how we can do some of these things, particularly in non-model species, the things that we all work on and the things that are really critical for conservation. So they're things like just being able to do gene editing, being able to then do assisted reproductive technology, something this audience knows an awful lot about. Again, that's perfected for some species, but then completely not at all developed for others. For marsupials, that's been a real challenge for us, is surmounting some of those ART challenges, and I'll talk a bit about that today as well. We also need to be able to do things like derived stem cells. And again, that's quite different for different species. We're very good at doing it in mice and humans, and we're not particularly good at doing it in other non-model species. And that requires a lot of time and effort to try and figure out exactly what those cues are to make stem cells in these other species. But if we really want to realize the potential of biobanks and do genetic editing and reintroduce lost genetics back into populations, we've got to figure out this stem cell problem for all species. We really need to figure that out. And then we also run a very large biobanking initiative here in Australia too. We set up Australia's first living cell biobank, which I'll talk about as well, which is another really critical resource that I'm very passionate about and think we need more biobanks. But the main species I'm going to focus on today, the one that my lab is really passionate about bringing back, is this one here, the Tasmanian tiger. So for those of you who have never seen this animal before, it was an incredibly unique marsupial. So it had a pouch and raised joeys in its pouch, just like a kangaroo. But I think everybody here can really appreciate just how much it looks like a canid, like a dog. It's got a very, very canid form. In fact, it's the best example of convergent evolution that we know of in vertebrates. And that's when two animals evolved to look incredibly similar. If you look at the skull of a Tasmanian tiger and compare it to the skull of a dog, you'd be really hard pressed to see any differences between them. They are incredibly, incredibly similar. But this animal sadly was hunted to extinction in 1936. The last animal, this one here, died in Hobart Zoo in September. Had a really tragic death. The keeper just simply forgot to put it away one particularly cold night in Tasmania, and the temperatures dropped below freezing. And this last animal, the last known animal of this particular species, froze to death in this pen. So really, really tragic story. But the thing that made this animal so important from a conservation perspective was that this was an apex predatory marsupial. And this was the only single apex predator mammal that we had on the landscape in Australia. And this is really unusual. If you think about the Americas, if you think about Europe, there are tons of examples of apex predators that you have on the landscape. You have bears and wolves and big cats, like all sorts of things that you have roaming across this landscape. But in Australia, we only had this one. This was our apex predator. And we know that those species play absolutely critical roles in maintaining ecosystems and ecosystem stability. So when you remove these animals from the ecosystem, we start to see huge destabilization of all of the species within that. And that has profound implications for conservation of all of those animals. And so there's a real imperative that if we have the technology, which we now do, to try and bring back some of these really important cornerstone species and put them back into these ecosystems. So one of the first things that we can see that's happened as a result of the loss of the Tasmanian tiger is that this particular species here, the Tasmanian devil, there was a massive upsurge in this horrific disease that this animal gets, and apologies for the graphic images here, but it gets this devil facial tumor disease. It's actually a transmissible cancer, a very unusual disease. And we can see by looking back at historic samples in museum collections that this disease was present in the Tasmanian devil population, but at a very low level historically while the Tasmanian tiger was around. But once the Tasmanian tiger was removed from that population, there's no apex predator anymore. There's nothing to remove these really severely diseased animals from the population. So instead, these animals persist, like the ones you can see up on the screen here, to very advanced stages of this disease. And while they're all feeding from the same carcass, and also part of their natural behavior is they bite each other's faces when they're all in a den together, they are transmitting that tumor from animal to animal. So when you have your apex predator there, it's removing these animals from the population and it's keeping this disease at a sustainable level. But once the Tasmanian tiger was taken away, we don't have that balance anymore. This disease has run rife. There's nothing to take those animals out of the population that have advanced disease. And so this is spread completely throughout the Tasmanian devil population now. This would have made that species go extinct already, but we were able to go and isolate uninfected individuals, bring them back into, clean areas, and they're being bred up in captivity and they are sequentially now released back into Tasmania. But because this disease persists there, they will all eventually contract the disease, and there's no sustainable way to have the population of Tasmanian devils in Tasmania without the Tasmanian tiger present. But they're just one single example, and we can see this imbalance happening completely across the landscape in Tasmania. It's not just the devils. There's been an explosion in kangaroos and wallabies, again, because there's nothing to predate those species, so to control their populations. And so that whole ecosystem is really starting to fall out of balance. And I'm sure many of you are aware of this incredible study that was done in Yellowstone where they removed the wolves. The wolf is the closest analog to a thylacine, actually. And so it's like removing the exact same ecological role from Yellowstone. And then they tracked, so that happened in 1926, and then they tracked it over the subsequent years, what was happening in Yellowstone Valley, as it's such a beautiful, pristine national park, that people were taking very good notes around what was going on in the landscape once the wolf was removed. And so, as you would expect, you see an explosion of the deer population. That's similar to our kangaroos and wallabies. So you have a massive explosion of that. They start eating all of the plants. So any new plant that's trying to sprout and grow gets eaten by the deer. There are no flowers or berries available anymore. So the bees leave the valley, the birds leave the valley. Eventually, the bears left the valley as well. Then the deer start eating all of the fresh growth that's alongside the river, because that's the most fertile area of ground. They muddy up the river, the beavers left the valley, and then in the end, the river actually changed its course through Yellowstone Valley. And they could see this catastrophic collapse of that ecosystem in Yellowstone. And so they made the decision in 1995 to put the wolves back in again. And within 10 years, that entire ecosystem had restored back to that natural balance, including the beavers returning and the river regaining its natural course through Yellowstone Valley. So it's just an incredible example how the removal of one species can profoundly impact not just the plants and the animals, but even the landscape. The shape of the landscape changes as a result of that. So we know that these animals, these apex predators in particular, have these absolutely cornerstone roles in maintaining these environments. And that's why it's so important to have them back there. In this case, luckily, you could just go to a neighbouring state park, find a pack of wolves and translocate them back into the valley. Obviously, we can't do that with the Tasmanian tiger because it's extinct. There are none of them left around. And there's no other marsupial that even comes close to the role that this animal played. There's no other animal that could step into that apex predatory role. And so we really have to think about, if we want to try and restore that ecosystem, we've got to be able to put that Tasmanian tiger back into Tasmania. So I think in terms of thinking about de-extinction and the kinds of species we want to bring back, this is one that really ticks all of those boxes for something that I think we can make a very, very strong case for. This was a completely human-driven extinction. We hunted them to extinction in a very, very short time. There's no replacement. There are no other species that look like it that can fulfill that role. It was a cornerstone species in that ecosystem, so it was very, very important for sustaining natural balance of everything else. And then importantly for us, it was a relatively recent extinction. So 89 years ago now, since that last animal went extinct. And so because it's relatively recent and Tasmania is quite a pristine, incredible place in Australia, there is a lot of intact ecosystem left there. And the food sources that it would eat have arguably become more abundant now in its absence. So it certainly has everything it needs, not just to survive, but also thrive again back in Tasmania. So we can tick all of those boxes. And the other unique thing I think about the Tasmanian tiger is because it was such an incredibly unusual and beautiful specimen, is there were loads and loads of samples collected for this that were stored in museum collections. And that's given us incredible DNA and even incredible RNA resources for this particular species. And these are some of the specimens that really made this possible. So we sequenced the first genome for the Tasmanian tiger probably about 15 years ago now. And we did it from a joey, a baby, a pouch young, a pup that was taken from its mother's pouch, which is that specimen you can see on the far left in the tall jar. And then they also collected that mother's head at the same time. So her head was removed and placed into a bucket of ethanol in the back of a museum collection. And we've also been able to get unbelievably good DNA resources from teeth. One of the nice things about teeth for us is you can actually drill into the tooth, which is what that movie's showing on the right there, into that soft pulp region. And you can get really pure thylacine Tasmanian tiger tissue, so it doesn't have any contamination. Whereas the head that you can see in the middle and then the joey that you can see on the left have been handled by 100 more people over the last 100 years. And so they have a lot of contaminating DNA all over them as well, which makes the things just a little bit more complicated when it comes to genomics. But one of the staggering things that we found is that middle specimen, that head in a bucket that has been sitting at room temperature in ethanol for the last 120 years, actually has unbelievably well-preserved, not just DNA, but RNA in the tissues. So by sampling that head, we've been able to actually look at gene transcription of taste. So we can look at the taste receptors in the tongue. We can look at the smell receptors in the nose. These are regions of the genome that are very, very difficult to annotate unless you have RNA, but we've got RNA from all of them. Obviously, we can also look at sight because we have the eye, we have the brain, so we can look at brain function and then muscles, bones, all the tissues that you can think of. It's really incredible that they preserved a head because it has so many different tissue types that we can learn so much about thylacine biology from. So really incredible. And so with all of those resources combined, we were able to sequence the first end-to-end or telomere to telomere genome for any extinct species that we've done now for the thylacine. So the Tasmanian tiger or thylacine genome is now better than most living animal genomes, just because we put so much effort into trying to rebuild that genetic code. And obviously that's our blueprint for how you recreate a species. So we've got to get that code right. So we've now been able to sequence through every single part of the genome, including the ends of the chromosomes, the telomeres, the middle of the chromosomes, the centromeres. These, again, notoriously very, very difficult parts of genomes to sequence. But because we have so many good resources, we've been actually able to piece that all together. And we can even look at things like the epigenetic profile across that genome as well. It's been so well preserved in these specimens. So really phenomenally able to completely rebuild that genome. So then how does this process of de-extinction work? Fine, we can sequence our genome of our extinct species and we've done that, but then what happens next? So what we do is we take that genome and we ask, once we have that complete genome, what is the closest living relative to our extinct species? So we just compare that genome to living animal genomes. And for us, that is a small mouse-sized marsupial called a fat-tailed dunnart that you can see at the top there. That is a lot smaller in scale to the thylacine, probably a little bit smaller than its foot. So really, really quite small. But when we compare their genomes, they're about 99.85% identical. So only 0.015% different. I say only, but it is a 3 billion base pair genome. So that still equates to 4.5 million differences. But the way that this technology works is you take living cells from your living animal, in this case, the fat-tailed dunnart, and then you go through using CRISPR, so our really advanced genetic editing tools that we have today, to just change all of those nucleotides where they're different, going sequentially through and editing all of those eventually 4.5 million edits across that genome. Once you've done that, you've recreated your thylacine genome, and then you can use our standard cloning technologies, ART, to turn that cell back into a whole living animal using things like somatic cell nuclear transfer, and they're techniques that we're working on currently in marsupials. And right now we're really focused on this DNA editing phase. So we have our established colony of fat-tailed dunnarts, we've got our thylacine genome, and we're going through and making all of those edits to recreate that thylacine genome. But while we're doing that, we're working on all of the assisted reproductive technologies to actually be able to turn that cell back into a living animal. Once we've actually got that cell and turning it back into a living animal, obviously we need a surrogate. So in this case, we would actually use the fat-tailed dunnart as a surrogate. So some of you might be thinking, how does this work if you've got an animal the size of a Labrador being born from an animal the size of a mouse? But this is one of the incredible things about marsupials is they have such tiny, tiny babies. And this is a dunnart baby on the day of birth, and it's about the size of a grain of rice. A thylacine baby Tasmanian tiger is exactly the same size. So there have been some collected at this early stage. And so what that means is even our tiny little mouse-sized marsupial can actually give birth to a baby thylacine, which is really incredible. They have only a 13-day gestation as well. So really short gestation and tiny, tiny baby at the end of it. It's still a lot of changes to make, so we need to figure out how we sequentially go through and edit all of these genes in the genome. And we've got a few clever ways of doing this now that we can actually scan genomes and identify regions that we think are responsible for giving them their particular form and function. And then using transgenic mice, we can actually test whether these genes are actually driving changes in their body shape and their skull shape. And so this is just an example of one of the regions we found in the Tasmanian tiger genome that looked like it was changing skull and limb shape. And so what we did is we just asked what this fragment does in a mouse, and you can see it's turned on everywhere the mouse is blue, in the face and in the developing limbs. And then we can actually put that piece of DNA into the mouse genome and ask how it changes shape. And we could actually change the shape of the mouse skull to start to direct it more towards a hypercarnivore. So we can start to learn how we can actually manipulate our genome to actually change the form of our dunnart to make our eventual thylacine. But one of the things I'm most passionate about is how we can immediately use all of this technology we're developing for de-extinction for conservation outcomes for marsupials right now. Because as I mentioned at the beginning, they're in this real desperate need for more conservation tools. So one of the first things that we needed to do was be able to harvest oocytes. If we want to develop this ART, I'm sure everybody on this call appreciates that you really need to have a good supply of oocytes to do that. We never had protocols for superovulation of dunnarts before, of any marsupial before. So there was nothing for marsupials in existence. So we have a really talented team of ART experts here who worked through at the arduous task of trying to figure out what that hormonal regime would be to get them to induce them to ovulate. And we get very good ovulation rates. I wouldn't call it super ovulated, so we call it very good ovulation instead. But we get a really good number of eggs. We probably get about 20 eggs per induced ovulation. They would naturally ovulate about 10, but we can then fertilize those eggs and we can culture them right the way through to blastocyst stage. So we know we're able to get really healthy eggs. And if we induce ovulate and get them to naturally mate, they'll produce live offspring. So we know that our induced ovulated eggs are healthy and good. And so we can now use this as a really viable tool for helping us develop all of the ART that we want to for marsupials. This act of doing induced ovulation also brings animals into heat, obviously. And so this has been already used for conservation in a lot of our wildlife breeding schemes. So as I mentioned before, the Tasmanian devil, on the brink of extinction, we really need to do something to try and save our Tasmanian devil population. But another species that's on the brink of extinction is the northern quoll, which is that little animal you can see sitting down the bottom of the screen next to the devil. But these are both related marsupial species, dasyurids. These are very feisty animals. If you introduce a male to the female and she's not receptive, she will kill the male. This makes them very, very difficult to breed in captivity, particularly when you're trying to maintain any genetic stock that you've got. You cannot run the risk of having one of your males get killed just because the female was not receptive enough. But using this induced ovulation scheme now, we can predictably bring that female into heat, introduce the male at the right time, they can mate and produce the litter, and the male doesn't have to die, which is great. Also, if the female loses her litter for any reason, we can use this to bring her back into heat again and get her to have a second litter within a season. So this is already being used for conservation and has been really pivotal in increasing the output from these captive breeding programs for these animals. Another thing we spent a lot of time on in my lab was actually perfecting how to make induced pluripotent stem cells. So we figured that out now for marsupials. Again, really complicated. One of the weird things about marsupials back on this slide is you can see that blastocyst on the far right. There is no inner cell mass. They don't actually form an inner cell mass. The embryo will just form on one side of that ball of cells. And so the signals that are required for making induced pluripotent stem cells were slightly different to what we see in mice and humans and other species. So we had to really unpack that. We've done that now. And once we could demonstrate that, Australia was finally going to get behind us setting up a living cell biobank, which we've done now at Museums Victoria, where we're trying to collect cells from as many marsupials, endangered and non-endangered, as we possibly can, and biobanking those living cells down now so that we never have to go through this process of de-extinction again. We won't have to re-engineer a genome because we'll have those living cells frozen down and we can go through that cloning process. And then the last example I'll give you of how we're using this is with the gene editing. So now that we can edit genes so well in marsupials, there are a number of different applications of that. The first one that we're doing is for this northern quoll that I said is critically endangered. This is actually the next species that is predicted to go extinct in the wild within 10 years in Australia. And the reason they're going extinct is because they eat cane toads. Cane toads come from South America. They are not native to Australia. They were translocated, introduced into Australia. They are toxic, they produce a poison, and none of our native wildlife have any natural resistance to that poison. But where cane toads naturally exist, there are a few mammals that can easily eat these cane toads and survive fine. So by looking at the genomes of those species, we were able to identify that it's just a single amino acid in a single gene. And in fact, it's just a single nucleotide in our 3 billion base pair genome that makes you either susceptible to cane toad toxin or resistant. So what we did is we obtained cells from our northern quolls. We genetically edited them to produce that cane toad toxin resistance allele. We just changed that single nucleotide, and then we exposed those cells. We turned them into stem cells. We exposed them to the cane toad toxin, and we showed that when we do that from that single nucleotide edit, we can make the quoll now completely cane toad toxin resistant. It had 6,000 times increased resistance to that toxin, which means it's completely resistant to that. Not only does this save the quoll from extinction, but it means that they can actually eat cane toads and help control this spread of this awful pest species across Australia. So it's a really nice example of how genetic engineering can be used in a conservation space to save our wildlife. So we're working now on perfecting that ART, that cloning process, so we can turn those cells now back into a living quoll and then start releasing them across Australia. So I just want to finish up by going, when I first started my career working in ancient genomes and, sequencing marsupial genomes and trying to understand how marsupials have evolved over the years, I would go to museum collections and open these drawers and think about how sad it was for all of the species that we had lost and had been collected and, are no longer out there and in the wild. But now when I see these museum collections, I know that we can sequence whole genomes from these animals. We can understand more about their genetic diversity, and we can actually engineer back in that lost diversity to recreate really, really healthy populations and resilient populations for our planet moving forward. So this is no longer a sad thing. I'm now so grateful that we collected all of these specimens that we can go back, we can re-engineer some of that lost diversity back into animals. And I actually think that's where de-extinction technology will have its biggest hit. I think there'll be a few species that we bring back through this whole de-extinction process, but the majority of the science will be used just to engineer lost diversity back into living animals so that we can make really healthy and sustainable populations of some of these, critically endangered species, which are down to just the last few individuals. There's no way they can sustain really healthy populations again. So hopefully I've shown you today that de-extinction science is really conservation at its core, and that we're developing these really new methods to conserve and preserve and then restore biodiversity, whether that's restoring biodiversity back into these living populations or bringing back entire extinct species and putting them back into the landscape. And just this last thought that, we've really changed the planet so quickly, and we're changing everything around our poor species and our poor ecosystems at a rate that they just cannot keep up. And we need to be really open-minded about some of these new tools that we need to put into our conservation toolkit to really help build resilient ecosystems again if we want our planet to survive this massive 6 mass extinction event that we're in currently and to be a sustainable planet moving forward. So with that, I just want to thank the team that you can see there and all of my funding, and thank you very much.
Okay. Thank you so much, Andrew. That was, you managed to to deliver a lot of key information in a very short amount of time, and it was very clear. I loved it. Thank you so much. So we have a lot of questions for you, and as we said, we're going to spend a little bit more time just for you because you cannot really stay for the whole webinar. So there are different types of questions. There are questions that are more technical, and then after that, there are questions that are a little bit more philosophical, but not necessarily philosophical, but like more like applications and translation of that knowledge. So let's start with first some questions that are a little bit more technical, and it's all about, okay, so you talked extensively about sequencing the genome and how complicated it was for the tyrosine, for example. But we have some questions about, okay, so once you are able to really sequence the genome, what about the methylation of the DNA and all the different aspects that where how are you able to trace back really the imprinting of the genes and all the packaging of the DNA?
Yeah, so I mean, there is some methylation data from some of the samples that we had, but obviously you've only got limited tissues there. But what we do know, thanks to the pioneering work done with somatic cell nuclear transfer, is that you can take a fully differentiated cell, right, put it back into an enucleated egg, and you will erase that epigenetic code, and then you reset that epigenetic code as those cells go through early embryogenesis. And they do acquire the right imprints. So if we take a marsupial imprinted region, for example, and put it into a mouse early embryo and let it go through that process, it will actually acquire the correct methylation. Where histones sit, what those histone marks are, and what those methylation marks are across the genome is all encoded in the DNA. So there are certain signatures that tell histones where to bind and then what modifications they should have. It's all there. So going through that process of early cloning can reset that. Obviously, that's one of the biggest challenges with cloning and why we see so many non-viable animals from that process is because it's very hard to go through that rewriting process. But there are methods now that they've really, really well perfected in mice just because it's easy, where you would take that cell, create an early embryo, let it get to the blastocyst stage, remove one of the cells from that. So it's already gone through one round of reprogramming and use that for another round of somatic cell nuclear transfer, let it go through again. And you can iteratively do this until you've completely wiped that slate clean and then let that genome reform what its natural imprint should be. So we feel really confident that you can actually get there. I mean, we know we can do that with a terminally differentiated cell type that, you know, a skin cell you can turn back into a whole living embryo by just erasing and resetting that epigenetic code.
Okay, thank you so much. Someone was, Jason Nguyen was wondering also, because you talked about, okay, DNA recovered from the skull or from the tooth, but if the only source of DNA is hair or fur, is it still possible to reconstruct a genome and is the information still accurate? Not decreased?
There's been massive advances in ancient DNA now. So from hair, actually, it's actually really good because there is hair actually, I mean DNA in the middle of the hair shaft. So they've figured out different extraction protocols now. So if that person's particularly interested, I would say look up Beth Shapiro's work. And she has done a lot of work in extracting really good quality DNA from hair of museum specimens that are hundreds of years old. The older the DNA is, the more fragmented it is, so the shorter the lengths are, and the more difficult it is then to rebuild the genome. So that can make it really challenging with just hair, but you can definitely sequence a genome with just hair. Whether you could do a really good genome build or not would depend on what quality that DNA is.
Okay. Thank you. Okay, so now let's talk a little bit more about the gametes. So how do you explain that everything is so complicated from eggs to sperm to embryos in marsupials? Why are they so special and so complicated to understand compared to other mammals?
I think because they have a different reproductive strategy, right? They really chose to give birth to very small babies and then have this prolonged period of lactation. So everything about them is quite different. But yeah, when you start digging into the things about marsupials, they have the longest sperm of any mammal. So they have these crazy long sperm that then swim completely differently. The egg gets this a shell coat as it's ovulated and travels down the oviduct that is almost impossible to inject a needle through. So we We have the laser. You cannot laser blast through the shell coat. It's impossible. We have to get these very special glass pointy-nosed pipettes and use piezo to even get the needle into the marsupial egg. Then the marsupial egg is really yolky, so it's really difficult to know exactly where we need to inject things, where we're putting it into the egg. But we are working through all of this. Again, from a conservation perspective, if we can do ART, if we can do AI, if we can do ICSI, like any of these techniques would have profound implications for our management of captive breeding colonies. So they're things we need to sort out anyway, but it's just lacked, the investment in really having people work on it. But hopefully we're making a huge amount of headway. I mean, just being able to do that induced ovulation has been a game changer for us, because now we can finally get enough eggs to start playing around with all of this work. But I hope within the next year or two, we'll have somatic cell nuclear transfer sorted, we'll have IVF or ICSI sorted, and able to actually produce viable embryos. But yeah, they're complicated in every single aspect. Nothing is easy in a marsupial.
That makes them so exciting, actually.
It does. Everything's so interesting. I mean, the fact that they don't form an inner cell mass is fascinating. It's just a completely different way of forming an early mammalian embryo, but then that made making induced pluripotent stem cells a nightmare for us. But, we learned so much when we figure these things out, because it is such a different way of approaching development, reproduction, the whole thing.
Okay, so following up on that, assuming that you can produce embryos from thylacine, can you elaborate a little bit more about the strategy about transferring those embryos to surrogate species, and what would be the candidates, and what kind of obstacles you are kind of foreseeing about that?
Yeah, so they would gestate within the Dunnart, so because they're so small when they're born. Then they could spend probably about 2 weeks in the mother's pouch before they would outgrow that. So they'd get that very, very early milk from the Dunnart. By the time we would transfer them out, they'd be about the size of a bean, like a kidney bean. That would be about them at 2 weeks old. And then we actually already have the milk that you need to hand raise marsupials, because we have so many marsupials that are hit on the road and are orphaned. And so we actually have this sequential milk formula that you can get from suppliers to actually hand raise even very, very small marsupials. So we're looking at making an artificial pouch that you could put them into. You could put them into a kangaroo surrogate pouch. The kangaroos are pretty happy with actually taking surrogate pouching in there, but that'd be complicated. It's more easy just to bottle feed them and put them in an artificial pouch. But obviously those first animals you wouldn't release into the wild, right? Like they would be quite artificial, their whole creation, but you would keep them in captivity in very large areas, and then you would hopefully be able to breed from those animals. That would be the plan, is that you would be able to make viable, reproductively viable offspring that you could start to then actually produce then your thylacines.
Yeah, and actually, that's kind of the next question is, okay, so now assuming that you get the first birth of thylacine, a breakthrough in science for sure, but how will you, I mean, not only you, but all the people working in this massive effort of the extinction, how will you make sure that you can scale up the effort, and then after that, well, repeat really the success, produce enough animals to really build a sustainable population that could be eventually reintroduced in the wild? So I mean, how are you going to be able to multiply that success, and where are the resources going to come from?
Yeah. So for the thylacine, that's a lot easier than it is for a mammoth, for example, right? I think we can all appreciate that that's a lot easier in terms of scaling up. You can have a colony of Dunnarts and have hundreds of Dunnarts that could be surrogate mums for these particular species. We also sequenced about 50 genomes so far of thylacine. So once you've got your telomere to telomere genome, it's very easy to do, just basic Illumina sequencing on any crappy sample that we can get from a museum, and you can get a really good genome from that because you've got something to map it back to. So we have a very good understanding of population diversity of thylacines right around the time that they went extinct, but then also back hundreds and thousands of years, so that we can engineer in, thousands of years of lost diversity if we need to. But we would start by creating a population that had that more contemporary diversity that existed around the time when they went extinct. So that's actually quite easy to engineer back in compared to re-engineering your whole thylacine. That's a much smaller amount of changes that you need to make to incorporate that diversity. So I think we can get diversity back in. I think for something like a marsupial, it's pretty easy to produce 50 animals, using surrogates. It's not that challenging. For some of the larger animals, that's definitely going to be a challenge. And then, for all of these larger animals too, they take decades to become reproductively active themselves. So, if you want to try and make them to start breeding to make a sustainable population, you have to wait even longer because they take a long, long time to become sexually mature and be able to breed themselves. So yeah, there are challenges. I think this is going to be not a quick process for a lot of species, but I think there are really critical species that we want to get back into those ecosystems before they fall apart.
Well, thank you so much, Andrew. I think we're gonna have to switch to the next speaker. But what we're gonna do, since you cannot again stay with us for the whole webinar, we can also kind of try to compile the questions that you were not able to answer with the name of the people, and then we can forward that to you because it's very exciting to see how much people are really kind of eager to know more about all the things that you are doing. So thank you so much, Andrew.
Absolute pleasure. Thanks for having me.
Thank you, Andrew. And it's really evident from the number of questions that have come in relation to your subject, just how interested the attendees are. So it would be great if we can keep the conversation going by forwarding some of those questions to you. Thank you very much.
Thank you.
Unfortunately, we have run out of time with Andrew, but our next speaker is someone who I'm proud to call a friend. Having met nearly 30 years ago when we worked together on an initiative that helped establish what has now become one of the world's leading commercial equine breeding and semen freezing services, Stallion AI Services. Tullis Matson is an entrepreneur. He's a charity founder, a conservation pioneer, a skydiver, and a proud dyslexic. He's dedicated to protecting global biodiversity, and he combines cutting-edge scientific technologies with bold advocacy to safeguard endangered species for future generations. In 2020, Tullis founded the charity Nature's Safe with a simple yet ambitious goal to transfer biotechnology from his company to the urgent challenge of halting and even reversing biodiversity loss. Nature's Safe is committed to preventing animal extinction by indefinitely preserving live cells from endangered species, and this is a mission that could change the future of conservation. So Tullis, I'm going to hand over to you. Tullis, would you like to share your slides?
Thank you. Can you see that there okay now, Matt?
We can, yes, thanks Tullis.
Brilliant.
Do you have your sound enabled? Just a quick sound check.
Yes.
Okay, great. Over to you.
Awesome.
Thanks, Tullis.
Great. Well, thank you, Matt, for that amazing introduction. And what a fascinating first presentation, I have to admit. So thank you very much for inviting me to this webinar. And yes, talking about some of the world that I work in, and I feel like I do have one of the luckiest jobs in the world, and I'm going to give you a bit of an insight to what I do. So the presentation title is Life After Life, and just showing some of the work. But my background, basically, I'm a horse person by trade. I've been breeding horses for virtually all my life. I've been freezing horse semen down. Actually, we started with Matt, nearly 30 years ago. So that's really where it started. So I'm a cryobiologist. This horse here is one that went and won the Olympics. This is what sort of pays the bills, you could say. But my passion is these ones at the bottom here, which are the rare breed horses. And we've had over 1,500 stallions come through our centre. And one of my ways is how can we save our rare breed horses and through using new technologies. I love new tech. And how can we develop that? And these are some of the rarest horses in the UK. And that's what we've started off doing. And this is what you're going to see a bit of tonight. We've got several companies, just all to do with about freezing things down in a way we can bring them back to life, or the cryo side, being a cryobiologist. And these are just some of them. We have a logistics company for shipping semen around the world. We have Gemini Genetics, which is the cloning side. Stallion AI Services, the foundation company. We do the same with dogs. So we collect and freeze dog semen and ship that around the world. And we're doing the same with livestock in the sense of freezing their tissue down. We're just about to start that and biobanking our livestock, but I'm really passionate about this charity setup called Nature's Safe, about freezing down species, which you'll see a bit later. I think it's always quite good to go back and look at the history of AI Quite often there's a different type of AI than we're used to talking about nowadays, artificial insemination rather than artificial intelligence. And the first references around the 1300s, actually, believe it or not. They actually used to put a sponge inside the mare, let the stallion cover the mare, get the sponge out, and with a press, they used to press the semen out, extract the semen, then they used to AI mares that way. So it's come on a long way since then. And then this foal here is one of the first foals back in 1955, was actually born by using frozen semen. So it's come on a long way even since then. This is one of the first videos that was taken in the 1930s when they're breeding horses for the German army. And this is how they used to collect semen from the stallions. They used to stallion cover the mare. The vet would turn up to try and extract the semen. Looks like followed by the postman, but no health and safety there. And then literally the veterinarian would go inside the mare and soup the which is luckily nowadays we have other techniques of extracting semen, but it's always quite good to see how they were used to do it. And any of you out there think you had a pretty hard deal in life with your job, just spare a thought for this person. I don't know what they got paid, but this is the first dummy mare, and luckily we don't do this nowadays, but if they had a great big stallion thundering towards this, I would feel sorry for the person sitting on that stall, and I hope they got paid a bit more than the minimum wage. But it's always quite good to see how it started, and I think this horse says it all. If you think I'm jumping on there, you've got another thing coming. But the dummy mares now are a little bit more sturdy and a little bit more easier to use, as you'll see in this video. This actual breed of horse is called a Suffolk Punch, incredibly rare. There's only about 80 or 90 left in the UK. There's only about 300 odd around the world. And we're trying to find ways that we can preserve them and freezing their semen down. And sometimes when you get inbreeding and you get the extinction vortex, quite often it gets more male gender heavy. So we want more females on the ground. And with these heavy horses, we looked into sperm sexing. We went to the cattle side, which again, Matt very much helped us out on, and we went and looked into them, see if we could sex some equine semen to see if we can get some more female sperm. And it had about a 95% accuracy, and this was the first foal, rare breed foal, born by this method. After 11 months of waiting, Ruby has produced a filly foal, and it's not just any old foal. This is a foal born by sex semen. We believe it's the first ever in the world for a rare breed animal to use sex semen. And using these technologies can really help this breed. There's only about 70 or 80 females left in the country. There's only 300 left in the world. They need all the tools of the industry, and just quite incredible now what we can do with science and technologies. And using this technology, maybe, just maybe, we can help stop these breeds from going extinct. But this holds the future. Little girl here. So that was quite a momentous occasion, really. There's about 11 months gestation period, and I must admit I've never looked between a horse's back legs to make sure it was the right sex before we did that film, and it was. And we've done quite a few more sex semen foals, and to be fair, they've all come out the sex that we wanted, which is great. And you'll see a theme when we go through this. Now this has been transferred to some more endangered species as well now. I think they're doing this work in Florida, and the sexing's been done in Texas. But it really just shows how it's moving forward. And if you can develop these technologies for the domestic side, then you can throw this across onto endangered species. We've been working with epididymal sperm, taking semen from the testes. We've been doing this for quite some time. When a horse had a tragedy and they asked, can we do it? And we gave it a go. And you see the semen on here, how it's totally immotile, or very little. And this is sometimes what semen can look like. And we add something to it. We call it SSPR, synthetic seminal plasma replacement. And this wakes the sperm up. And now we do this, we've done about 2 this week. I've got another 2 more to do next week. And we do 2 or 3 a week epidydymals, not just equine, on other species as well. So this is sort of a last ditch saloon. If they haven't preserved the genetics, there is other ways of doing it. This bull was bought at the sales, and after about a week or so went and broke its leg. And the guy, vet, said, oh, there's some guy down in the south of England that can actually extract semen. I've never done bull's testicles, so I went onto Google and looked at the the anatomy of the bull's testicles. Well, you can imagine everything came up then, but we had a go. And then we had the first calf on the ground by this method back, I think it's about 2015. And now this, off that one procedure, they had 30 offspring from that one procedure. And now this bull on the right-hand side was this calf. And that really led us on saying, crikey, we can use these technologies, and we've thrown this across now onto other species as well, as you'll see a bit later. So quite often it starts on the domestic side and really throws it over. But travel the world, really setting up labs, teaching people how to do this cryo work. We've had a very busy few years. I think it's something like 14 or 15 different countries in the last year or so, setting up laboratories in India, Japan, Finland, Iceland, and now the Faroe Islands. The Faroe Islands, if you're not too sure where it is geographically, it's between Scotland and Iceland. It's a population of about 50,000 people, or maybe even 30,000. And they've got a horse there called the Faroese horse. There's only 97 of them left, 30 stallions, and they're so inbred. I mean, they've got an inbreeding coefficient of over 30%. So we've been out there last year in 2024, collected, and we've been back again this year, and we've collected off all 30 stallions. And I think this is maybe one of the first times in the world that we've collected a whole breed of animal, the horse, and biobanked it basically. And we're hoping to go out now and take tissue from each one of these and bank them down as well next year. These were quite wild, some of these horses, but they seemed to, the beauty about them, they had good libido. So they were jumping on the mares, and now we've actually trained them to jump on dummy mares as well. So, which really, really did help. But the beauty about them, we used to have to go and quite often catch them from outside. But generally they were very willing to please. Sometimes the Faroese weather isn't the best, and the odd stallion we actually had to go and try and collect outside, which was always interesting, as you'll see this next video is.
Right, our next collection is actually outside. I don't think I've collected in weather like this before outside, but we've got the stallion there, the mare's coming up the track here, DJ's in the truck with the AV, but yeah, pretty amazing surroundings here, but yeah, pretty raw weather out here, but let's see how it goes.
They were quite brilliant. I mean, as you can see, the libido is great. There's not many animals out there that you could do it in a gale force winds and they still perform. So, but it was incredible. And we got the semen from one. They've never been AI'd. Everything's been natural covering. So we AI'd our first mare back in 2024, which was one of the first ones. Well, it was the first one that's ever been done. And then this year we've the mare foaled. So this is the first foal born by artificial insemination. This really showed what these technologies can actually do to a breed of horse that is could be facially on the brink of extinction. And then we can translate that across to other species that we've actually worked on as well with camels. This wasn't necessarily on a rare breed side, but this is just trying to further technologies. Camel semen is very difficult to freeze. And they've got the beauty camel and the racing camel. I usually ask a question, which do you think is more expensive? But usually comes back to quite often the beauty camels are very, very expensive out there. So we're doing some work as well in how we can freeze camel semen as well. Other work we do is genetic preservation, freezing tissue down in a way that can be used in the future for cloning purpose on a domestic side. So we've got a company called Gemini Genetics, and this has been running for 7 or 8 years now. And we do cats, dogs, and horses. And the work is actually done in America with Viagen, but we actually freeze all the tissue down in the UK and then ship it out there. This is one of the original clones here. You can see Gem Twist. He was a gelding, so in other words, he couldn't breed. So they decided to clone him, and the one on the right is the clone, and that's the one we have in our yard behind me. And he is a stallion in his own right, and he's done exceptionally well. And he's basically, you could say, a sperm bank for the original. That just shows what these technologies can actually do. This is one of the ladies in my life. She's amazing. She's called Gem. She's a clone dog, and she's actually by my feet now. So when we maybe finish this presentation, you can all have a look at her. But she's a clone dog. She's one of the top. Used to be a clone of one of the top field trial dogs in the country, and this we went up Snowdon, one of the highest peaks in the country. So it just shows what is possible out there. We thought, well, if we can do these technologies to all these equids, why can't we transfer this over onto other species? So this is where Nature's Safe was born. We're facing the 6th mass extinction of all time, between 50 and 100 species a day, and sometimes nearly going extinct. It's quite frightening. By the time we've all gone to bed tonight, or some of you getting up, in these next couple of hours, another 15, maybe 20 more species will have gone extinct, which is could be really, really frightening. So our mission is to save by collecting and indefinitely storing reproductive and cells and cell lines from species from all over the UK and the world. And this, if I was to get rid of all the slides, this says it all really. This is from Revive and Restore. It just shows that, all the species going into the tank, and some of the work we can do on it, which we heard a bit before in the previous presentation about gene editing or reproductive technologies, but a lot of the technologies haven't actually arrived yet. But what we do know, if we don't put them in the tank in this end, we might have nothing out the other end. So I think it's imperative that we bank as many species as we can before it's too late, because some of these species are going at an alarming rate. And you'll see at the bottom here, or when these species were preserved. But we collect the samples, and we've got up to 5 days from when an animal passes away to get those samples in, and we get the reproductive cells and the skin cells and the ears and the and we tend to get ovarian or testicular tissue, eggs and sperm, and skin samples. I'd say 85% of the samples we get in are skin, just because it's easier to get to. With sperm and eggs, we really need those samples within 12 to 24 hours when we're freezing them down. One of the fastest declining species on the planet is coral. Believe it or not. We've lost 50% of our coral since 1950. They are the lifeblood of our seas. And I was actually at the Horn Museum only last week for the 2nd time. This is the first time we went there. Coral spawn once a year, roughly 2 weeks after a full moon, 2 hours after sunset. And you can see the coral spawning here. And they release them in bundles. There's sperm and egg together. And then they go to the top, and then we separate them out. The sperm come away from the eggs, and then we can collect the sperm at the bottom.
It's absolutely incredible. This is coral sperm. You're looking at this under the eye sperm. Look at those sperm go. I wish some of the other species would have stuff as good as this. And we can actually really...
And that just shows, it amazed me how good the semen was on some of this coral. There's a 3D printed device here that we did, and we've frozen cryovials in filtered seawater with DMSO, and going down at 20 degrees a minute. And then a few days later, they thawed that out and mixed the frozen semen into the eggs. And you can see the sperm this side. Look at the tail on that. I mean, it's absolutely incredible looking at these semen samples. And we got the first cryo coral babies in Europe, which was absolutely amazing. So we feel like it's absolutely amazing what this science can actually do. And again, coral is very, very important. They're the lifeblood of our seas. I've watched this video maybe about, 100, 200 times. It always gets the hairs at the back of my neck going up. These are the last 2 northern white rhinos on the planet. There's none left, apart from effectively they are still alive, but they're virtually on the brink of extinction. But this is what Richard Vine had to say.
Hello everybody, my name is Richard Vine and I'm talking to you from the Old Pettit Conservancy in Kenya. Behind us you can see the last 2 remaining northern white rhinos on the planet, and we are doing our best to recover this species from the brink of extinction. Both are infertile, and therefore artificial reproductive techniques are the only options that are open to us. Nature's Safe is also playing a critical role in saving the world's biodiversity. by creating one of the first living tissue banks on the planet. Their work is incredibly important, and we look forward to working with them in the future.
I mean, that's just such a sad video, and it's, you know, us as a mankind, we've, you know, produced that scenario, and we've got to try and make sure, you know, we try and halt the extinction process as much as we can for many of our species. It really is very sad to see that video. So the science behind it, as you may know, there's lots of biobanks all over the world, but there's very few what's called living biobanks. So there's lots of dead biobanks where they're freezing at -80, and those cells with no cryoprotection, those cells rupture and break. So the cells are non-functional, but the DNA is still there intact. But where our cells are all alive, so we're freezing in a living biobank, and that is the difference of what we do. We take the skin samples, usually the ear, because those cells regenerate much easier for us. But there are other parts that we are freezing down. The beauty about this, you know, if an animal dies on a Friday, Saturday, Sunday, even Monday, they can ship those skin samples to us, and it's got the whole DNA of the animal in. We can freeze that down and for 10, 20, even 1,000 years and help bring that species back if it's on the brink of extinction. Our data and record keeping are absolutely paramount. It's only as good as obviously storing these samples if you've got the data package behind it. And we use the Matcher system. I know Matt's involved with this, but this is something that's really showed when people look into the charity who run some of the big benefits of that. We've got a really good barcoded system with the whole identity of that particular animal on those cryovials. And, you know, when you've got a few hundred or thousands of samples, you can get away with those systems. But when you've got thousands of millions, maybe hundreds of thousands of samples, it's absolutely imperative. A system like this can actually make such a huge difference in how you store your samples down. And this just shows some of the, you know, this was on the New Scientist last year, saying the northern white rhino, could it be saved from extinction literally by frozen skin cells? So this is just showing, you know, some of this work and what it can be done. This was a rhino that passed away in 2019. We just froze the ear down, part of the ear of the rhino. And then this year we got the ear out of to the -196 nitrogen tanks. We thawed it out and started to get the cell lines growing. After a few days, we had a few hundred, and after a week, we had millions of cells. And the beauty about this is you can replicate. You can replicate. When you have sperm samples or in straws, once those straws have been used, that's it. But you have an infinite supply when you're regrowing these cells up. And this was highlighted this year on the BBC as well, just showing how so important these technologies are to helping save some of these species that are on the brink. And as it was alluded before, it's called the extinction vortex. When you get these really low numbers of species and they're virtually in this vortex, Nature Safe could come and actually using that genetic line and bring that species and to help it climb back out of that extinction vortex when that gene pool is so small that you can bring back those lost genetics, and it really can make a difference. When it comes to epididymale sperm, I say it's not just the horses and the cows. Yeah, we do it on all these other species now. And this was a cheetah, very inbred. Don't normally have the best quality semen again because of the inbreeding, but this just shows what it can be done. It passed away. They sent the testes into us and we froze it down. I don't know whether you've heard of something called the mouse deer, one of the smallest deer in the world. The testicle weight is 1.3 grams. Incredibly difficult to extract the semen, but we did. And we actually managed to get some good quality semen from that. The mountain chicken frog, incredibly rare. I think there's only a few hundred left in the world. And we do a lot of electron microscope with the pictures and get these amazing pictures to see. And we're trying to build something called a sperm atlas to try and get some really good pictures with this. We don't know whether this is a normal good sperm or not, so we need to build up that data. Avonsera, and when you do this work, a lot of the time it doesn't always work. Sometimes someone has maybe never even frozen from Avonsera, for instance. So we have these spreadsheets that are building more and more and more. We've got hundreds of different species on our spreadsheets, and we're learning all the time. So when it doesn't work, we alter it. But this is one of the main successes. We froze it down, thawed the semen the next day, and the semen was incredible. So not only have we preserved that animal's genetics, we've we've got a protocol which is just as important, if maybe even more important. So if this work needs to be carried on in the future, those protocols in place. And building up this sperm atlas can really give us a good intel whether this semen is good, bad, how it relates to that particular species as well. And so this is something that we're building on, and we're just about to send a load from a snow leopard, I think, and some other species we're about to send away and look at their semen quality and looking at how that semen looks as well. Work that's even been done on snakes, collecting semen from snakes. So there's no species that we don't want to be preserving. And this is working with the African lion safari in Canada. And they perfected this collection technique. And we've been helping them with the cryopreservation side when there's going to carry on trying to do some more work with this, because there's very little done on this side of things. Let's say preserving skin samples. The other technology is cloning we talked about, and obviously using nuclear cell transfer. And this is one of the first rare breed, horses to be produced by this nearly 40 years ago. They've saved a tissue sample from 40 years ago to produce this foal. Quite incredible. And back then, they may never even thought that it'd be used for cloning. So preserving tissue can have so many different pluses. The black-footed ferret virtually went extinct in the 1970s and was cloned and was brought back because the genetic pool was so small. Someone had the foresight to preserve skin samples from 30 years ago. So this really just shows how important it is to preserve these animals before it's too late, before they're gone. I think it was briefly spoken about in the last presentation, but obviously with his skin samples, you saw the cloning, but there's other future technologies that will be coming around in turning a skin cell into an egg or into a sperm. And obviously this has been done on the mice and a few other species, but it's something, those future technologies that we should be really looking at. Our vision is to build up hubs, and we would love to hear from anybody out there on this webinar. If you're interested in partnering up or learning more about this, we're about knowledge transfer. We cannot do this alone. It's all about coming together with these technologies. This is a crisis we're finding with many of our species, and we've got to leave a legacy behind for future generations. I think if we don't, they'll look back at us and say, you have the ability to do this, but you did nothing. So it's so important that we actually forge ahead and preserve these species. This is something I love and enjoy, in setting up hubs and biobanks around the world. And yeah, we're calling out for your help, and we'd love to hear more from you if you're willing. This is some other work that we did in South Africa. This is me. Part of my job was hanging on to an elephant's penis for about an hour a day. But it sounded a bit weird, but actually it was quite incredible. We did 7 bull elephants over a week. We collected semen from them, tissue samples. And again, we've lost so many elephants over the years, and they're in big game reserves, which is amazing, protects them. But again, they can inbreed again. So we need to be collecting. So far to date, we have collected from 366 different species just in the 5 years. But that is obviously a drop in the ocean of what we got to do. This is our cryo store here. As you can see here, we have over, nearly 1 million straws in store of equine semen, but also it's where we store all these other species in these cryo vaults. We've just been nominated for the Earthshot Prize, which is quite fantastic. We just learned about this literally 2 weeks ago. So if anybody don't know, this is one of the big Earthshot Prize. It's once a year. It's £5 million given out to 5 great causes. So fingers crossed for that. But we need your help. It doesn't, obviously we need funding, but we're always looking for people to come on board. So if it's something you're interested, please reach out. These are some of the people that really do help us. We are a charity at the end of the day. And I think we all know this person, the great Sir David Attenborough. And he, in one of his last programs called The Mating Game, I've spoken to you now for just under 25 minutes or something. And what he's summing up in these 3 sentences or 4 sentences is what sums up the whole lot. By using these current technologies, if we so wish, we have the ability to save any animal or at least save its tissue until we discover how. And I think that's a very sort of fitting ending to us. And I thank you for listening. I'd love to hear some questions as well. And thank you, Matt, for. And there's some details if anybody wants to take a quick picture of that before it disappears. And you can reach out to me on any of those social handles as well.
Tullis, that was a fascinating presentation. It's great to see how you're endeavouring to transfer assisted reproductive technologies already developed in domestic species to conserve more of the critically endangered species. In the interest of time, we're going to move on to the next speaker, Tullis, but there are lots of questions coming in, so there will be time at the end to get more involved in a Q&A session. So I'm going to hand back over to Pierre. Thank you again, Tullis. Pierre is going to introduce the next speaker. Thank you.
Yeah, thank you. So last but not least, Andres Gambini. Ciao, Andres. How are you doing? Good to see you.
Pierre. Yeah, good to see you too.
Okay, so Andres is right now in Australia, but of course he's not from Australia. He's a lecturer at the University of Queensland. But he comes from Argentina, where he did his PhD in 2000. He finished his PhD in 2008. And actually, the title of his presentation is kind of a brief summary of his CV, because he's been working on all those different species. He's been the one really making, producing the first clone horse, in South America, in Australia. So that's he has a lot of things to say, but. Now that he's in Australia, he's also working. He's not going to talk about that, because he's also working on marsupials. And last year, they produced the first IVF embryos of a kangaroo, which is also a huge achievement. So, Andres, you use many different advanced assisted reproductive technologies, and the floor is yours. And welcome.
Well, thank you so much, Pierre, for the invitation to the webinar, and thank you everyone that is joining us today. And I think my talk today will cope really well to the previous two amazing talks that we just had, because I'm going to be talking about horses and other members of the equid family, such as zebras and donkeys and mules, and how we can apply assisted reproductive technologies, and more specifically, different ways of producing embryos for animal production, but also for conservation. And if I have time, as Pierre mentioned, but only if I have time and Pierre allows me, I will briefly comment and give you a snapshot of the work that we have done in marsupials this year, and also in the saltwater crocodile. So why I'm here today talking about this, and Pierre mentioned a little bit on it, but I'm a veterinarian originally from Argentina, and I had the opportunity to do my PhD in horse cloning back in 2010, and we were able to produce the first clone foals in South America that were born in Argentina, and I've been working with this technology pretty much since then. Another technology that I've been working on is ICSI, and OPU, so ovum pickup combined with ICSI, and I'm very sure that all the audience is very familiar with this technology, and we were able in our lab, again in Argentina, back in 2018, to produce the first foal in our country produced by OPU followed by ICSI. And at that time, I also started collaborating with Australia, where I'm based now. I have a position at the University of Queensland now, and I'm doing teaching and research in Brisbane. But we started a collaboration with Melbourne University, and we were able to produce the first clone foal in Australia. So as you can see, I've been producing embryos in horses for a very long time now. And I just wanted to show you how this technology that when I started was not really well established, but now OPU and ICSI, they are both established technologies in domestic horses. OPU and ICSI is now we probably have more than 40 laboratories around the world providing the service of OPU and ICSI in horses. In Europe, the number of embryos that are being produced in the lab through ICSI is higher than the number of embryos that we are conventionally flushing from the mares. And in terms of cloning, we pretty much the same. We have now at least 7 commercial companies that have established. Many of these companies are actually in Argentina and in Brazil, because these are the countries that we can have access to a lot of ovaries from slaughterhouses, and that facilitates cloning. And there are at least 3,000 clone foals that have been born. The numbers are, of course, really hard to estimate, because this is commercial work and the numbers are not always told. So just to show you the workflow of what we are doing right now in Australia, in Queensland, we have our embryo lab in Gatton campus, and we have connection with industry partners that they collect oocytes from mares and, of course, semen from stallions, and they send the gametes to the lab. In the lab, we can produce embryos, and these embryos are then frozen and sent back to the industry partners that they will perform embryo transfer. And this is not new, right? And you're probably very familiar with this if you're a human embryologist or an embryologist in general. But what I want to take 1 minute today is to have this amazing opportunity to reach a lot of people that is working in the human IVF world, to show that I am seeing a huge opportunity on using the horse as a translational model for understanding more the impact of human-assisted reproductive technologies. We have now a commercial ongoing OPU ICSI programs all over the world, and we have the opportunity to use the mare as the model to understand more female infertility. And there are many things that you can see in the slide that are just ideas where we can explore and do research. But in my mind, aging, for example, is one of those things that we can collect a lot of data from the OPU ICSI program that we have in horses, that we can potentially take messages from the human IVF world. And that's because we are collecting oocytes from mares across all different ages. Very similarly, the stallion can potentially become our model for understanding male infertility. And there are a lot of similarities, and we are using and trying to apply this technology to stallions that have some degree of infertility, and looking at OPU and ICSI as a solution for that stallion. And again, plenty of ideas that we can combine. Something that I believe is truly amazing as an opportunity is that we are creating athletes, we are producing offsprings, and we are monitoring these offsprings. So again, a huge opportunity to contribute on how producing embryos in the lab is impacting that long-term and the multi-generational impact of assisted reproductive technologies. And lastly, something that is amazing for me as an embryologist as well is that most of the things that we are using nowadays in an equine lab are from the human IVF lab. So we are using the same vitrification protocols that are being used for human. We're using the same culture media, same incubators, same needles, and so on. So again, I wanted to take that 1 minute to show how the opportunity that we have now here with horses. And just to give a few examples of what we're doing in the lab, not super rock science, but we're using laser technology, for example, to select and immobilize sperm, as you can see here in the video. This has been done in humans already, but there's not a lot of studies looking at potentially the impact of this method of sperm selection, and if it is something that we should be implementing more or not. another very beautiful example on how we can contribute from the horse into the human assisted reproductive technology is the fact that this year was born the first horse through maternal spindle transfer. This is a technology that it's already applying human, but there's a lot of debate around it. And we have now an opportunity to use the horse as a model where we have a lot of mares that are infertile, that we cannot get embryos through OPU and ICSI, and we can potentially innovate using this type of other nuclear transfer technologies. But I'm not gonna keep talking too much about horses today because I want to spend most of my time in the other members of the family. And one of these members is donkey. I'm fascinated about donkeys myself, and a lot of the people will, think that a donkey is pretty much very similar to a horse. And yes, they do belong to the same family, but the common ancestor was more than 4 million years ago. So they are two very distinct species with very different behaviors and characteristics. But we have these two domestic species as part of members of the equids. But they are not the only members. We have more species that belong to this fascinating family. And this is where the zebras come in place and the wild donkeys come in place. So we have 3 different species of zebras and 3 different species of wild donkeys. And this is just to make it easier for the audience today. But what is really concerning is that, as an example, one of the species of donkeys, the African wildlife, is considered critically endangered right now. There are less than 200 mature individuals in the wild. And there's clearly a need of taking action. And this is where we believe, as it was said in the previous talk, that applying assisted reproductive technologies that we have developed for domestic animals into wildlife, it's what the contribution that we can do. Similarly, in the group of the zebras, there's one species of zebra that is also endangered right now, listed as endangered, that's the Grevy's zebra. And I'm going to show you the work that we have done to try to contribute to develop assisted reproductive technologies for zebras. And lastly, as it was said, again, in the last talk, the Shabasky force, another member of this family, also considered endangered. And in fact, cloning was already applied for this species. But what it makes this group of animals super interesting is that we can mix and match everything, really. So probably you are very familiar with the mule, which is the cross with the mare with a jack, that's a male donkey. But we can also do the other way around and produce a kine. So this will be the offspring of a jenny that we cross with a domestic horse, with the stallion. And this is just, if you think about it, an amazing opportunity to understand and to study genetics and epigenetics and the maternal environment and so many other stuff that offer us the members of this family. And similarly with the zebras, we can mix zebras with horses, and we're going to have live offspring. That's a source. And we can mix zebras with donkeys, and we're going to have a sonkey. So yeah, it's just fascinating that these intercross are producing viable offspring, although most of the offspring will be sterile, infertile. But what is even more interesting is that within the members of this family, the genetic plasticity that we have, it's just amazing. Researchers, few years back, they demonstrated that if we took, for example, a zebra embryo from a zebra, so it's 100% zebra embryo, and we transfer that zebra embryo into a mare, into a synchronized mare, that mare can get pregnant and can deliver a healthy zebra foal. And the same thing we can do with donkeys and mules and zebras and horses. So it's a fascinating aspects of the family that allow us to have the opportunity to have and use domestic equids as recipient animals for wild equids embryo. So I'll now go straight into applying the technologies that we have been hearing about and talking about in the last two lectures. So we have the opportunity to partner with one of the zoo here in Argentina when I was working here as a researcher. And I got a call from a friend that was working there, and she mentioned that a zebra have just died. And I said, just send me the testes and send me a piece of skin. And that's what we did. We were able to isolate zebra sperm from the tail of the epididymis and freeze that sperm using our knowledge on horse semen freezing. And with the skin cells, we were with a piece of skin, we were of course able to isolate fibroblasts, skin fibroblasts, and freeze those fibroblasts. What we did next was use the zebra sperm and injected that zebra sperm into a horse egg. And this will be producing source embryos in the lab. And with the skin cells, what we did, we used a horse egg. We enucleated that horse egg, we removed the DNA from the horse, and we introduced and we used the somatic cell from a zebra as a DNA donor for that oocyte to then produce an embryo that will have the genetic of a zebra. So let me tell you a little bit of the findings of this research that we did. And the first thing that we do in our lab, we like a lot to do ICSI. Pierre, you know this. And one of the things that we do is we can, of course, with ICSI, as you know, you bypass all the barriers of fertilization and you can inject any sperm into any oocyte pretty much. And that's what we did at the beginning. We used pig eggs because that was this easy to have access to pig eggs. We in vitro matured these eggs, and then we injected horse sperm. And we knew that when we injected horse sperm into a pig egg, we will trigger that initial oocyte activation and initial steps of embryo development. So we can see the two pronuclei here, for example, and the two polar bodies. And when we injected zebra sperm in pig eggs, we were able again to see that we can still have pronuclear formation, although the rates were a little bit lower compared to horses, they were still happening. So we decided to keep moving and then inject that sperm now, that zebra sperm, into a horse egg. And this is a bit of our table with the results. And I'm not going to spend too much explaining the numbers, but if you look pretty much, of course, we have a horse control here. So this is injecting horse sperm into horse eggs. And this is the blastocyst rate when we inject a zebra sperm into a horse egg. So it seems to be a bit low, but of course, this was just one animal. And we pretty much demonstrated that what we are seeing in vivo can be replicated in vitro. And we can produce embryos in the lab, these hybrid embryos in the lab. What happened with the cloning? So with the cloning, one of the technologies or the innovations that we developed during my PhD was embryo aggregation. So what we did, we produced zebra embryos. All these zebra embryos are coming from the same cell line, the same animal, so they are all genetically identical. But after doing the cloning, we removed the zona pellucida. So what you can see here are 3 embryos that they don't have zona pellucida and they are under a time-lapse machine. And we culture the embryos together in the microwell, and we do aggregation of 3 embryos. So basically, this embryo aggregation technique allows us to create an embryo, one single blastocyst that it's coming from 3 embryos officially. So this, we published a lot around this technology, and we demonstrated that embryo aggregation improves embryo quality, pregnancy rates, and cell differentiation, and so on. I can give a full talk on embryo aggregation, but yeah, I'm gonna stop the video because basically this will reach to the blastocyst stage, but the video is a bit long to spend all the time on it. So what we saw is that compared to horse cloning, that we were using a cell line from a horse as our control group, we were able also to produce zebra blastocysts in that cloning rounds. And it looks like cloning zebras it's easier or more efficient than cloning horses. It might be, but if you work in cloning, you know that there's a strong impact on the cell line that you're using. So it's not something that we can officially say, but you can look at the blastocyst rate, and we were amazed on how easy it was to obtain blastocysts when we were using a horse enucleated egg to reprogram that zebra skin fibroblast. But we decided also to look at the embryos more in detail and understand whether that horse ooplasm was doing a proper reprogramming of that cell. And we did that by looking at some markers at the blastocyst stage. And you're probably familiar with this. SOX2, which is a marker of the inner cell mass, and then YAP1, which is a marker of the tropoectoderm cells that, of course, are going to be more placenta. And then we compared clone horse embryos with zebra clone embryos, and we pretty much didn't find any difference, suggesting that a horse ooplasm can reprogram a zebra skin cell and contributing to understand even more the genetic plasticity that we have within the members of the family. So now, just for the sake of time, I'm gonna move on with donkeys. We were able to achieve the first donkey IVF embryo in 2022. We reported that. Doing embryos in donkeys is not as easy as it is in horses. We had to set up the ovum pickup technique. This is a work of one of my PhD students here in Argentina at the University of Rio Cuarto. But long story short, we were able to successfully collect oocytes. And in animals, compared to humans, we always collect immature oocytes. So we then need to do in vitro maturation. There are a lot of things that we learn when we move from one species to another species. We're looking at how OPU impacts the estrous cycle, the fertility of the donkeys, and so on. But what I want to spend maybe a little bit more of time, it's in this next slide, because we are now thinking on a domestic donkey as a potential model to understand more about aging and assisted reproductive technologies, because donkeys compared to horses are more resilient animals. They live longer. So we are looking at, for example, mitochondrial activity in donkey oocytes that are young and donkey oocytes that are old. And we're also, of course, comparing that with horses. And we're using different dyes to look at the mitochondria and whether that mitochondria is active or not. And just to summarize a bit of our finding is what we are seeing is that all donkey oocytes seems to have a total number of mitochondria within the egg. And of course, that kind of gives us a little bit of more total number of active mitochondria. So the ones that are healthy, which were the red before. But when we look, of course, at the ratio of the, what out of total mitochondria, which one are active, still all donkeys, animals are not, eggs are not able to compensate from a healthy young donkey oocyte. Anyways, this is the contribution ongoing that we have in donkeys. We needed to set up in vitro maturation. We realized that in vitro maturation in donkeys is way longer than in horses. We still need to know if this is because the media that we have developed is not yet 100% fine-tuned for donkeys, or if it is that just naturally donkeys will take more time to extrude that first polar body, as you can see there. And then we decided to do some ICSI, some intracytoplasmic sperm injection into donkey oocytes, and we were able to obtain the first donkey blastocyst produced in vitro. And we're now producing more blastocysts. We are doing that comparison between young and old donkeys. And of course, we're also having our mule control group. So if you think about it, producing a mule in the lab, it's injecting a donkey sperm into a horse egg. And this of course, for us, it's very useful to have as a control for that sperms, to be able to measure the sperm impact on development. And so far, we are not having like huge difference across all these different hybrids and species. We're also doing some time-lapse imaging work and looking at the morphokinetics of the embryos, but I don't have time to go into the details of that today. So just to kind of wrap up my talk, I think I'm close to the time. I'm hoping that I have provided some evidence that we now have a beautiful model that we can use to understand more about human-assisted reproductive technologies, and that domestic horse-assisted reproductive technologies, that we now have shown that the technology can be moved from other members within the same family, and that we can pretty much apply this technology to those other members of the family that are critically endangered, as I showed at the beginning. And I don't know, Pierre, if I have time to show something about the kangaroos.
Okay, you have 1 minute. Go ahead.
Okay,
of course.
I'm going to skip acknowledgement then, and then I'm going to just jump into the work that we did, and we published this early this year, and that was producing the first kangaroo IVF, but it's not officially an IVF, it's an ICSI embryo. I'm just showing you here a beautiful slide with some of the pictures of how a kangaroo egg look like. This is an ovary first. We are collecting ovaries from dead kangaroos, and then we collect the oocytes from the ovaries, in a very similar way that you will do this with cats or with mice. And what you can see here, these are cumulus oocyte complexes from kangaroos. And then we had to track down in vitro maturation for this species, because as Andrew mentioned, marsupials are completely different. But we did it. We were able to in vitro mature kangaroo sperm, where you can nicely see there the first polar body. And then we injected a kangaroo sperm that we collected from the epididymis and that we freeze. And we were successfully on producing embryos that cleave. And then we just fix and look at DNA quality of the embryos. We're also doing something similar in koalas. These pictures right here, these are immunofluorescence on koala sperm. Koala sperm is fascinating. They have 12 different type of heads. There's a lot that we can learn from understanding biology of marsupial. This is a koala ovary, and one of my PhD students is developing koala oocyte recovery, and we just published a paper now where we recover for the first time oocytes from koalas. And we are looking at basic stuff, just as how the normal oocyte in koala should look like. And just, to have fun, and because I'm in Australia, I couldn't help myself. And we did some crazy research on far North Queensland in Australia with saltwater crocodiles. I'm talking about 3.5 meters animals. And we were able to collect semen from cloacal massage from these animals. And we are also now working on preservation technologies and chilling media for developing and helping assisted artificial insemination in these species. All right, that's it. I'm going to go back to my acknowledgement questions, but I think, yeah, thank you so much for everything.
Well, thank you so much, Andres. That was really great. So we're gonna switch directly to the. Everybody's gonna come up on camera, and we're gonna switch to the Q&A session, and we're gonna request our speakers, our guests, to really provide some quick and snappy answers to a question, because we don't want to be too long. But again, the most important is that we have questions that really show that there is a huge interest from the audience. I would say that first of all, Andres, I'm going to start with you. You demonstrated pretty well that the horse could be a very good model for human reproductive medicine. And there is a question about actually that is related to that, is because apparently it's very difficult to superovulate equids in general. So is that true, by the way? And does it create kind of a potential challenge if you went for your work in IVF or ICSI?
Yeah, that's a good question. Yeah, it's superovulation in horses is challenging because of the anatomy and histology of the ovary in equids. So it's been tried many times, and it's not something that it's commercially viable right now, and the efficiency of the superovulation is just not going anywhere. So that's a big difference with the human IVF workflow in domestic species. In pretty much all domestic species, we need to if we want to collect a lot of oocytes, we need to collect those oocytes from immature follicles, so antral follicles that are growing. That being said, we can still recover an in vivo mature oocyte. We can inject hormones in the mare to induce that pre-ovulatory follicle to ovulate, but we will only recover one. And that's why the technology has now moved into just aspirating all the follicles that we can see in the ovary and doing vitro maturation instead of trying to collect in vivo mature oocytes.
Thank you.
Thank you. I have a question for Tullis, actually. This is a question that relates to conservation of species, and it's fairly evident from your talk that it's almost a race against time to proactively cryopreserve as much biodiversity as possible. But the question is, if the world fails to act fast enough, what class of animals or species lost do you fear will have the greatest ripple effect on ecosystems, and why?
Thank you, Matt. I mean, to me, there's one answer here. It's insects at the end of the day. Yes, we're doing all these the mammals and that side of things, but the insects, the pollinators, we need them. And, without them, our whole ecosystems and the biodiversity of our planet can fall apart. So the insects, and they're very difficult to cryopreserve. And so we're actually going to be looking at butterflies, trying to cryopreserve butterfly larvae for I think one of the first times. That's a PhD student we're looking at. We've lost 80% of our butterflies in the last, I think it's 40 years, which is frightening. So our insects are a challenge, there's no doubt about it. And I think if we were to lose them, we're in serious trouble.
I suppose it's less glamorous as a class of animals because they're not, fluffy and cuddly and large, but they're probably, as you say, we often forget the importance of them and how they are really integral to the stability of ecosystems.
Absolutely. And the rhinos, the elephants, and all these amazing other species out there, there's a lot of work focusing on them, but the less fluffy animals, as you say, there's less work on them, but they're vitally important.
We better get on with it. Thanks, Tullis.
Okay. A question for you, Andres, and it's going back to more like about the. Can you hear me? I'm sorry. Looks like my internet is getting slow. Okay. It's more about the cloning, and it's a question from Valeria Cañada. So how genetically identical are cloned animals to their nuclear donors, particularly considering de novo mutations and mitochondrial differences? So I don't know if you can get a snappy answer on that one, but
yeah, I'll try. But that's a very, very very, very good point. When we are doing cloning, we need to be considering that we are putting a somatic cell that has its own, mitochondrial DNA within a egg that will have a complete different pool of mitochondrial DNA. So we are definitely changing the mitochondrial DNA to that nucleus, and there's a lot of evidence that there's a strong connection and communication within the nucleus of the cell with the mitochondrial. So that's definitely impacting the cloning, and more. We really need to pay attention to this when we think on cloning as a strategy for conservation, right? Because we don't want to mess up things. We want to try to create embryos and future animals that we know that are genetically valuable for the population, and just changing mitochondria is not the way to go. So there's a lot of other technologies that we can apply to replace the mitochondria later on. So it's not impossible, but that's a really good point.
Thank you.
Thank you. Another question relating to cloning, which we've had from a number of attendees in relation to biodiversity. So if biodiversity depends on genetic variation, can cloning ever truly support it? Or does it merely buy time in a race against extinction? And that's a question for either of you, as you're both involved in the practice of cloning.
I think the classic case in this is the black-footed ferret. We saw that, some people say cloning, how does that help biodiversity? If you clone lots of one particular animal, no, it doesn't. But if you bring in a lost genetic line back that has been lost for history, and that biodiversity of the genetic pool is very, very small, that clone coming back can really help climb out that extinction vortex. And the black-footed ferret was a classic example of that, of bringing a species back that died nearly 40 years ago to help a species that's here now. And that just showed what it can do.
Integration with other species.
Yeah.
Okay, thank you.
Yeah, just to comment on that, I think that's one of the beauty of cloning, right? It's a machine to copy genetics, but if the genetics that you are copying is a very valuable genetic that was lost, or that a genetic that didn't have time enough to contribute to that population, then you can put it back, and then you can officially increase genetic variability within your population. And that's what you need to save a species that is at the brink of extinction. You need genetic variability.
Thank you. There's another question from Valeria, actually, regarding another form of assisted reproductive technology, which is lyophilization, freeze drying, I think. So have either of you worked with sperm lyophilization, particularly regarding DNA integrity preservation and subsequent embryo development? Do you see any potential for this technology?
Yes, I can quickly comment on that. We are indeed working with that. I have one of my PhD students. His PhD is based on freeze drying horse and donkey sperm. We are looking at DNA fragmentation levels, and we're looking at how other important proteins within the sperm changes with freeze drying. We're doing proteomics on that. So we'll soon give you a lot of more information about it. But what we can see, and of course, Pierre has a lot of background on this as well. But it's a very good way to keep the DNA intact. Of course, the sperm will die, but the sperm will remain intact. Yeah.
Yeah. Pierre, do you have a view on that? As you have some knowledge of that?
Well, yes. I mean, we don't necessarily work on lyophilization. We work on desiccation or dehydration here. We don't use the same techniques because lyophilization is supposed to have first a freezing process and then sublimation of the ice. But what we do is we dry directly, and actually, Andres spent some time in our lab earlier this year to work on domestic cats and other types of endangered species. But I agree with what Andres is saying. It's a very intriguing and cool options, but we still need to do a lot of basic research because, of course, there are obvious applications to human reproductive medicine, but we need really to be careful about, you know, the consequences of the preservation and the stress of the cells, you know, during dehydration and all this kind of stuff.
Wouldn't it be wonderful though if we could end up in a world where there was no ultra low temperature storage and we just add water like coffee? That would be fantastic. Right. Thank you.
Okay, Matt, you had a question again about the success rates of cloning.
Yes, it was a very quick question. You know, could you give us an indication as to what are the success rates that you could expect from cloning in domestic species? And that's again to either of you. Perhaps Tullis, do you have any experience of that?
Just with it depends what you call success rate. When the tissue samples come in, yes, if we get them within usually 5 days, the success rate is incredibly high. I mean, it's over 95%. It's very high. And we get those cell lines to grow and freeze them back down when we ship them off to Vigen. I know the success rate's been incredibly high. If you're looking, obviously, at the embryo failure rate, that's different. I don't know those figures, to be fair, off the top of my head. So, yeah. Andreas, you might be able to.
Yeah, I can comment a bit more on that side. Of course, now that we have many commercial companies that are not necessarily publishing, you know, the new changes from the technology, the technology is officially a little bit higher, potentially in a commercial world than we have in science. And the efficiency will change in different species. So different species cloning technology has different efficiency. In one of the species that we're doing a lot of cloning, which is horses, I can comment on that. And we are probably having nowadays a 30% blastocyst rate with cloning. So the blastocyst rate efficiency is quite high. The pregnancy rates are a bit of the problem, not the early pregnancy rates, but those pregnancy that are able to keep going until term. There's a lot of pregnancy losses within the process, and the main issue is the placenta. So what we really know is that in clone animals, the placenta does not develop properly. So there's a lot of work trying to improve the trophoectoderm or the placenta development in clone animals. That's will definitely improve the efficiency of the technology. And something that is important for the audience is to understand that cloning relies a lot from the somatic cell. So there are some cell lines that are very good, and you create a lot of blastocysts, a lot of pregnancy, a lot of live, healthy foals, and some other cell lines are just not giving you anything ever. So there's something that we don't really necessarily know in depth that is making that huge difference.
Yeah, and this is living proof. Yeah, of what is possible.
Oh, so it does work.
It does work. Yeah.
So much opportunity to develop and evolve the technology. It's the prospects and the future is looking very bright, and the proof of the pudding is in the eating, as we can see in the arms of Tullis.
Okay, so I think it's time to thank everyone and to close our webinar. So, of course, I'm going to start by thanking you, Tullis, and you, Andres, and we thank also Andrew, even if he's not with us. Thank you to the audience, and I would like to thank, of course, Matt for his help to co-pilot this webinar. So, thank you so much.
Likewise, thank you, Pierre. And we'd also like to recognise the I3 initiative and the organising committee, Jacques Cohen, Peter Naz, Marianne Cervantes, Thomas Elliott, Fran Farley, Violetta Sura, and of course, Giles Palmer. Thank you very much, guys. It's a wonderful initiative, and we really look forward to what's to come in 2026. So on that note, please do join us again next year to talk about many new hot topics and the latest from the IVF world. If you are celebrating, then please have a wonderful, happy and safe festive season, and we wish you health, wealth and happiness in 2026. Thank you, everybody, and thanks again to the presenters.